Namaskar. A very warm welcome to the online portal MOOC, that is Massive Open Online Course on Human Genetics. Module 31B, Human Genome Project and its implications. The main objectives are to understand about the 1000 Genome Project, to know about the development of 1 lakh Genome Project, to know about the Genome Asia 100K project, and to know about the implication of human genome projects. Introduction. After the completion of Human Genome Project, many new genome sequencing projects were started globally. The technological advancement and applications of genome sequencing become exciting. The contemporary model systems, sequencing of prokaryotes, plants and animal systems provided enormous comparative data. The scientific community accepted the challenges and implications of genome sequencing to address a variety of issues. Hence, in this episode, the following aspects are going to be emphasized. A. 1000 Genome Project. B. UK 1 lakh Genome Project. C. Asian 1 lakh Genome Project. D. Indigenous Genome Project. And E. Implications. A. 1000 Genome Project 2008 to 10. The 1000 Genome Project was started in 2008 with an international research effort to build the most detailed map of human genetic variations. The primary goal was to create a comprehensive catalog of human genetic variations, namely SNPs, CNVs and indels. Secondary objectives were the support of better SNP and probe selection for genotyping platforms in future studies and the improvement of the human reference sequence. Furthermore, the completed database would be a useful tool for studying reasons under selection and understanding the underlying processes of mutations and recombination variation in multiple populations. Organizations involved in the project are the Beijing Genomics Institute, China, the Wellcome Trust Sanger Institute, UK, and the National Human Genome Research Institute, USA. The new participants are 454 Life Sciences, Applied Biosystems, and Illumina Inc., USA. During its production phase, the 1000 Genome Project was delivered sequence data at an average rate of about 8.2 billion bases per day, which is the equivalent of more than 2 human genomes every 24 hours. Mainly, the 1000 Genome Project has different phases that are assigned for various sequencing works. Pilot phase. This phase of the 1000 Genome Project was carried between 2008 and 10. The map of genetic variants with detailed data is published in 2010. The 3 projects of the study include the low coverage whole genome sequencing of 179 individuals of 4 different populations, high coverage sequencing of trios, and exon targeted sequencing of 697 individuals of 7 populations. The map was created from the sequencing data and identified around 15 million SNPs along with their locations, frequency of alleles and the haplotype structure. This map also included the small insertion deletions called indels and about 20,000 of other significant structural variations, including copy number variations called CNVs and presence absence variation PAVs. All the information from the study is expected to be the basis for the next phase of research. The variation seen in the genome can be of two types. The first, which can be rare variants and show the severe phenotypes of the simple traits, and the second type can be the more common variants and show the mild effect. All these variations are tried to address by the 1000 Genome Project. Phase 1. This study was started in 2009. This method included the low coverage exome sequencing of 1094 individuals from the 14 different populations. The genome mapping showed approximately 36 million SNPs along with 2.8 million indels and 14,000 deletions. Phase 2. This study was started in 2011. This method also assigned for the low coverage exome sequencing of 1722 individuals from 19 populations. Phase 3. This final phase of the 1000 Genome Project was continued until 2015. The sequencing of the 2500 individuals is done with the creation of a new variant catalogs. Along with the sequencing, which is used to map the genetic variants, transcriptomes, the study of gene expression to form the mRNA, was also studied under this 1000 Genome Project. The main goal of this project was to provide a detailed catalog of genetic variations in the human genome, including SNPs and other structural variations. The genetic variations will give rise to the study of different diseases in the research field. These genomes are sequenced using the techniques of next generation sequencing. This project aimed to study the variations and provide the results of the study to other researchers as publicly accessible resources. B. 100,000 Genome Project. This project was announced in 2012 by the Prime Minister of the UK. Genomics England company, owned and funded by the Department of Health and Social Care, was set up to sequence 100,000 whole genomes. The main goals are to create a transparent and ethical program based on consent, to bring benefit to patients and set up a genomic medicine service for the National Health Service patients, to enable new scientific discovery and medical insights, and to kickstart the development of a UK genomic industry. This project is mainly concentrated on rare diseases and aims to provide personalized medicines to the NHS. Sequencing of 100,000 genomes from around 85,000 of NHS patients was the task undertaken by this project, and finished the sequencing by December 2018. This project covers the main objectives was to bring an understanding of the genetic variants which can lead to different treatments and diagnoses. It also addresses the importance of genomic medicines to the public. Now, the researchers are studying the help of genomics in the medical field. It can be easy to interpret the data from the study and treat the disease. This project tried to solve the problems of occurring rare diseases including cancer in the UK. The main reasons to select the rare diseases was the event of the death of about 1,60,000 people by cancer in 2011. The number of cancer cases reported every year with new cases. Genomics has the potential to deal with rare diseases and cancer conditions because both diseases are linked to the genome. This project, which was completed recently, has uncovered new diagnoses and improved treatment for patients with rare inherited diseases and cancer. In time, there is the potential of new and more effective treatments. This makes the UK the global leader to integrate genomic technology for routine health care. This rich data set can be used for the next generation of precision therapies, diagnostics and clinical actionable insights. See Genome Asia 100K project. Genome Asia 100K is a non-profit consortium hosted by Nanyang Technological University Singapore and 3 biotech companies, namely Macrogen from South Korea, Genentech from the USA and MedGenome from India, USA. This was launched in 2016 to understand the genome diversity of Asians by sequencing 1 lakh genomes living in Asia. This is proposed to catalog genetic variation, population structure, disease association and founder effects. First, it is aimed to sequence 10,000 Asian individuals for ethnic stratification and 90,000 individuals will be sequenced later. The whole genome's reference data sets will be developed from 7 global regions, 219 population groups and 64 countries across Asia. This data with clinical and phenotype information will help the more in-depth analysis of diseased and healthy individuals. D. Indigent Genome Project. In April 2019, the CSR Institute of Genomics and Integrative Biology, Delhi and Center for Cellular and Molecular Biology, Hyderabad, initiated the 20,000 Indian genomes sequencing project. The objectives of the project are to enable genetic epidemiology and create public health technologies applications using population genome data. Whole genome sequencing of thousands of individuals representing diverse ethnic groups from India, predictive and preventive medicine with a faster and efficient diagnosis of rare genetic diseases. This investigation is aimed to understand the genetic diversity on a population scale, make available genetic variant frequencies for clinical applications, epidemiology of genetic disorders to enable cost-effective genetic tests, carrier screening applications for expected couples and enable efficient diagnosis of heritable cancers, and pharmacogenetic tests to prevent adverse drug reactions. Implications. 1. HGPs help to start the new ENCODE, that is Encyclopedia of DNA Elements consortium, to identify the non-genic elements in the genome, such as the promoters, inhibitors, enhancers and other functional structures which control the gene expression. 2. The HGPs have helped to start another international consortium, that is Human Proteome Organization, with the goal of identification of the structure and functions of the specific proteins encoded by the genes in the human genome. 3. The HGPs have provided many new tools to analyze and share the data of the work worldwide, which can be easily accessible through the internet. The sequence databases and gene banks are available for research and a variety of applications. This has helped to develop the supercomputers and high-end technologies to obtain the targeted outcome rapidly. It has benefited the commercial development of genomics research related to DNA-based products, a multi-billion-dollar industry. 4. The HapMap analysis has enabled the identification of several common and rare diseases associated with genes. This has helped us to diagnose the disease by genetic screening and gene testing more accurately. The outcome of the Human Genome Project helped identify the disease risk to the physiological response to medications, which has led to personalized medicine. The advances in DNA sequencing technology provide individuals with a low cost of sequencing the whole genome and the inexpensive diagnostic test. Therefore, the genome data can help us understand diseases, including genotyping of specific viruses for treatment, identification of mutations in different forms of cancer, and complex diseases to design more accurate medications and their effects. 5. Comparisons of specific DNA sequences of humans with the closest living relatives, the chimpanzee, reveal 99% identity. From comparative genomics, we know that the genome of modern humans arose between 150,000 to 300,000 years ago. Study indicates that populations of Homo sapiens deviate from Homo neanderthalensis ancestor population as recently as 370,000 years ago, and genome studies have suggested that there was no interbreeding between them. Therefore, genome data helps to trace the origins of the human genome. 6. Law and social sciences. The human genome sequence information has a profound effect in the field of forensics for correct identification of individual from a minimal amount of biological samples, such as a few hairs, a spot of dried blood, or semen or saliva. The DNA fingerprinting saved many innocent men and women from the wrongful convictions for crimes they did not commit. The establishment of DNA banking from all convicted criminals helps for the identification of perpetrators of future crimes. Comparative DNA sequence analysis of mitochondrial DNA and on the Y chromosome DNA have confirmed Africa as a cradle of modern humans. These markers have also identified the waves of migrated humans from Africa for over 60,000 years ago to populate the other continents of the world. DNA sequence variations help trace the roots of humans and identify the relatedness of one human population to another. 7. The human genome's information has advanced the studies that have identified the genes and specific sequence variations that contribute to disease risk and a multitude of traits. This has helped healthcare professionals to practice predictive medicine and preventive medicine. The research on pre-symptomatic genetics has enabled countless people to live longer and healthier lives. The mutation responsible for certain cancers like breast, colon have been identified, and with the early diagnosis these cancers can be most effectively treated. 8. Ultimately, the human genome sequence data holds benefits from genomic medicine to human evolution. To conclude, every genome project has its contribution to the sequencing of genomes. This helped researchers to find out the genes, variations and various essential factors in each genome. The 100,000 genome project was proposed to find rare diseases, and the main goal was to provide personalized medicine. Understanding the genetic variations, population structure, disease association and founder effects are the significant issues of the Genome Asia 100K project. Therefore, genome projects are ultimately aimed for better human health and welfare. Thank you.